Imagine being able to see every color of the human genetic spectrum, all in one panoramic view. Welcome to the world of the human pangenome. This is Double-Stranded Podcast, and today we're joined by Cristian Groza, a bioinformatics Ph.D. student who's trying to illuminate the hidden and variable regions of our genetic tapestry. Get ready for a deep dive into the heart of what makes us, us. Could you clarify the concept of a pangenome and how does it differ from a conventional reference genome like the GRCH38?
Right. So in the word pangenome, we have two things. We have the first part, the pan, and the second part, the genome.
Mm-hmm.
Everybody knows a bit about the second part, the genome, which is the DNA sequence of an organism or one individual. The word pan means that now we're talking about the sequence of everyone, right? So we're no longer talking about a single genome. We're talking about the genomes of every individual of that species, essentially. And so far GRCH38, for example, is really only one genome. It's not all genomes.
Mm-hmm. So the paper mentions that the draft human pangenome contains 47 phased diploid assemblies. I was wondering, how were these specific individuals were selected for the cohort? Like, what's the selection process?
Right. So the HPRC selected these individuals in order to maximize genetic diversity.
Mm-hmm.
If we look into their paper, we see that the majority of individuals are of African ancestry. Then following that, the second largest subpopulation are individuals of American ancestry. And then we have a smaller group of individuals of Asian ancestry.
Mm-hmm.
So this was done in order to maximize the number of variants, of genetic variants, that will be observed by sequencing the smallest number of people.
Mm-hmm. And why is that? So so you're saying that these ancestries have the most genetic variation?
Right. So we know from previous studies, from HapMap, the 1000 Genome Project, that individuals of African ancestry are the most diverse.
Mm-hmm.
So their genomes are more dissimilar from each other than, for example, two individuals of European ancestry or of Asian ancestry.
Mm-hmm.
And that's because the African population is really the oldest population on Earth, and they had more time to accumulate mutations and genetic variation. While the European population and the Asian population, well, they diversified more recently after humanity left Africa, which is why Europeans and Asians are less diverse.
Mm-hmm. And what about the sex? Because we have XX and XY, so is this something that they consider as well?
I would expect that the male to female ratio to be about equal.
Mm-hmm. They managed to add 119 million base pairs of euchromatic polymorphic sequence and 1,529 gene duplications relative to the existing reference GRCH38. So before I ask my question, can you please first define euchromatic polymorphic sequence and gene duplications?
Right. So the genome has a particular structure. It has these regions at the ends of chromosomes that we call telomeres.
Mm-hmm.
They are composed of the single motif that's very short and repeats over and over and over. And then in roughly around the middle of chromosomes, we have what is called a centromere. Again, this is composed of repeats, so a small number of motifs that repeat over and over again. And this is not euchromatic sequence, right? This sequence is usually in a repressed state. The chromatin is very compressed and compacted, and usually is very poor in genes. So we do not call this to be euchromatic sequence. Other regions outside the centromeres and telomeres for this study is called euchromatic sequence.
Mm-hmm. What about the gene duplications?
Right. So the human genome has about 20,000 genes. They found that in some of these genomes, certain genes appear in more than one copy, or in a larger number that is present in the reference genome. So some individuals might have one copy of a gene, while others might have two copies of a gene. I'm not sure if these are like full copies or functional copies. They might not be, but that's what they obtained on a first pass in annotation.
And so like I don't fully understand like what's the process. They just like sequence everyone and then like somehow average out and like put out something as like the average.
Right. So the number you quoted is probably the total across all the 47 genomes. If you look at per individual basis, it's probably something like 100 duplicated genes per genome.
Approximately 90 million of the additional base pairs come from structural variation. So maybe you can tell us like what is a structural variation?
Right. Well, in human genomes, we observe multiple classes of genetic variation. Many people know about the single nucleotide variants, which is the substitution of a single base pair from A to T or from C to A. Also, we can have bigger mutations, for example, indels, which can involve anywhere from one base pair to 50 base pair. But as soon as an indel or another sequence mutation exceeds 50 base pairs, we're starting to think of it as structural variation because it comprises a larger segment of DNA.
And like, what's the importance of structural variation in the study of genomics?
Structural variation is important because it involves many base pairs and can have larger effects. Until now, it was very difficult to access structural variation. So technologies in the past could only sequence very short fragments of DNA, 100 base pairs at a time mostly, which gave us access to the small genetic variations such as single nucleotide variants and the indels, but it did not really allow us to resolve the sequence of structural variation. So in this study, they used better technology, long read technology that can sequence larger fragments of DNA in one go. So they are able to give us the sequence of these structural variants, which until very recently was very difficult to do.
So, well, since my project is related to GWAS, I was just curious. So, basically, like no matter how many structural variants they just find, it doesn't like really affect me because like I'm just concerned about SNPs. Is that correct?
Well, it certainly can affect you. So in the pangenome, there might be SNPs that are not present in the reference genome you are working with. So it will be possible for you to find those SNPs and include them in your project. Moreover, you could even do GWAS on the structural variants themselves. So genetically speaking, a structural variant or a SNP, it's just genotypes. So there's no reason why you can't take the structural variant genotype and plug it into your GWAS models and obtain associations with the phenotypes and so on.
Yeah. Who would like directly benefit from Pangenome, like an updated version?
Well, of course, the benefits are not well known yet because it's such a new development, but areas like precision medicine, where the genetic background of individuals might be important. For example, there are regions in the genome such as the HLA or the KIR, which are very genetically diverse, and they were poorly represented by the reference genome because it only shows us one possibility out of the many possibilities. So by using a pangenome reference, we would have a fuller picture of what is the reality and maybe make better decisions.
So the paper states that using the draft pangenome reduces errors when discovering small variants by 34%. Could you discuss the methodology that led to this significant improvement and why it's significant?
Right. So when we analyze genomic data, we usually take a whole genome sequencing data set, and to understand it, we have to align it to the reference genome. But again, this data did not come from the reference genome. It came from an actual genome, a real individual that is different from the reference genome. So this difference between the actual genome and the reference genome leads to a phenomenon called the reference bias, right? It's like taking a sentence and trying to look for it in a book that doesn't contain it, right? You will make mapping errors, and this will decrease your accuracy and decrease your sensitivity when doing variant calling, for example. So the pangenome fixes this. It sort of gives you many versions of the same book. Some versions of this book do contain that sentence that you are trying to find. And when you find it, well, you find the variant, and this increases your sensitivity, which allows you to find more mutations.
So the detected structural variants per haplotype have been boosted by 104% compared to GRCH38 based workflows, and by 34% compared to using previous diversity sets of genome assemblies. So how will this boost in detection impact genomic studies, and how might it affect our understanding of diseases with a genetic component?
Any genetic disease that is due to a structural variant will benefit from this approach. Structural variation is particularly difficult to detect when the haplotype that is presented in the reference genome is very different from the actual haplotype in the individual, because you don't have to detect the variant. You kind of have to rediscover a very large segment of the genome. This was not possible before with short reads. It's beginning to be possible now with long reads, and to truly achieve this task, you really need to do genome assembly, where you don't just resolve small pieces of the genome, you resolve the whole genome, and then you can do a full genome to genome comparison to actually find all the ways that the genome can rearrange itself.
So when in making a pangenome reference, what are some of the greatest challenges faced during the creation of this draft human pangenome?
Right, so there are multiple challenges. The first challenge is actually obtaining very high quality assemblies. To make a pangenome, you don't need just any assembly. You need a reference quality assembly. That means that the segments of DNA, full contiguous pieces of DNA, have to be very long, preferably comparable to the size of whole chromosomes. And this is not really an easy task. We are talking about telomere to telomere assembly. The first attempt was really in 2001 when we were sequencing the first human genome, and that took billions of dollars. Today, it can be done with a few $1,000s. So this is the first problem that had to be solved. So in HPRC, they solved this problem, again, 47 times. The second challenge is to find a way to represent all these genomes. It's not enough to just generate assemblies. You have to take these assemblies and relate them to each other, right? We have to be able to take a genome annotation, something that tells us where the genes are and where all the biologically relevant regions are, and find them in the new assemblies. Right now, the proposed solution for this is something called the graph genome, where we take all the parts of the genomes that are the same in everybody and we collapse them into a single copy, because we don't need to show them twice if they're the same in everybody, and keeping the parts that are unique between individuals as distinct segments of DNA. And then we take all this data and relate it together using edges, right? So this creates a sort of a data structure that looks a lot like a road. So every genome in our data set will be a path in this road. So right now you can look at a sort of map where there are multiple ways to get from A to B, and all the ways from A to B describe a possible genome. And today we call this a graph genome.
So if I'm not mistaken, they only had 47 like their sample size was 47, right?
Yeah.
So like why don't they do it for like 47,000 people?
So the answer is really money and resources. Their goal is to reach 300 genomes, if I'm not mistaken. Of course, this will have diminishing returns at some point. After you sequence a few hundred genomes, the amount of new variation that you discover becomes smaller and smaller, right? The genetic variation in the human population is sizable, but is not infinite. So at some point, we will stop discovering new variation that is biologically relevant.
So I'm just asking this because like I have no clue. But like let's say in some part of the world I do whole genome sequencing on someone, and I just find like a novel mutation. So can I just like submit this to be added to the reference or something?
Well, of course you can take the new genome that you have and sort of add it to the pangenome yourself. So today the tools exist to do that. However, the point of the pangenome is to be a stable reference resource. So it's unlikely that everyone will be able to add their own genome to it. There will be a heavily curated set of genomes that is very high quality and well maintained. And people will be able to take that resource and sort of integrate it in their own research. And one of those ways to integrate it is to add your own genomes to it and see how they compare to that pangenome.
Yeah, nice. Because I was thinking that maybe like I will just build a dictionary of all the mutations, and like everyone, they can just submit their mutations, and just like this would be like the biggest collection of all the seen mutations in human genome. Do they have like a project like this, or just?
Right. So this sort of application already exists in the form of databases. So today we have databases of SNPs that relate these SNPs to all sorts of descriptions. Like, for example, if they are coding mutations or non-coding mutations.
You mentioned personalized medicine earlier. So what are the potential applications of this pangenome, like specifically for personalized medicine, and like especially in the field of genetic diseases?
Right. So in my most recent work, one way we applied the pangenome is to try to diagnose rare diseases in children. The challenge for us was to take a large set of structural variants that is present in every genome, around 20 to 30,000 structural variants, and reduce it to a much smaller set of structural variants that could be curated manually by a clinician. So the way to do this is to take the common variants and throw them away and only keep the very rare variants. And because they're the most likely to be pathogenic. And we use the pangenome to get an estimate of the frequency of every structural variant and the filter for those that are very rare, like 1 in 100 individuals or less.
So would you say that you are among the ones that actually benefit from a better pangenome for the sake of your own research?
Well, my whole research involves correcting the reference bias I was talking about earlier in epigenomics. So I was exploring the ways in which using a pangenome instead of a reference genome allows us to identify the epigenomic state of structural variation and other variations such as indels. So yeah, I believe that personally I benefited a lot from this, especially in completing my PhD thesis.
Yeah, they mentioned that the assemblies cover more than 99% of the expected sequence. What does this remaining percentage represent, and how might it be tackled in the future?
To date, even with the improvements in the sequencing technology, there are still very difficult parts of the genome that cannot be resolved. Several examples include regions like the centromere and also regions of the acrocentric chromosomes that have very complex, repetitive sequences that cannot be fully spanned by the sequencing technology. So in the future, this will probably be addressed with even longer read sequencing technology. For example, a nanopore could probably be used to span these difficult regions. Of course, right now, nanopore has a high error rate, but we can still correct these reads with other data, such as high fidelity long sequencing reads or even short reads.
Any additional comments about the human pangenome?
So far, HPRC did the challenging work of creating the pangenome. However, in my experience, I found that other researchers that are not directly involved in this effort still have difficulty of working with this resource because the tools are not developed yet. So if you are a genetics researcher, you probably use IGV a lot or the UCSC genome browser to understand your data and to carry out your research. Shockingly, there is no such equivalent right now for the pangenome. So if you are a researcher that studies biology, who makes use of reference genomes, but you are not a computational genomics researcher, it might be very difficult for you right now to make use of the pangenome. So I hope that in the future, other people will develop the tools that are necessary to make the pangenome a really useful resource.
Great. So last question, what's next after this draft pangenome?
So the following steps include increasing the number of assemblies that are part of the pangenome. Since you've read the paper, you probably noticed a figure where they show the rate at which new sequences are being discovered as we add more genomes to the pangenome. And in that figure, it's obvious that after the last genome is added, the rate of a discovery of new sequences is not zero. In fact, it's nowhere close to zero. So from that modeling, they expect to discover a total of 600 million base pairs of achromatic sequence.
Wow.
And so far, they found only about, if you remember the number.
I guess 119 million base pairs.
Right, so they're 1/6 of the way there.
Yeah, thank you so much.
Thank you, I had a lot of fun.
Alright, I really hope you enjoyed this episode. If you did, please leave us a great review on your favorite podcasting platform, and I'll see you again next time.